Thank you for joining us. This is the Atypical Alzheimer's Disease theme session. What is atypical for Alzheimer's disease? From syndrome to biology. Before we start, we have some housekeeping. For those who'd like to get continuing education credits, there's a desk on the far left of this room. We've got a stellar lineup of different speakers, bring expertise from different disciplines. In the interest of time, I'd like to make a start by introducing our first speaker, Deepti Pucha from Massachusetts General Hospital.
Okay. Good morning, everybody. Thank you for coming. I'd like to just get started by thanking the conference organizers and thank you to the FRS organizers for putting together this session. I will kick things off now by talking about memory impairment across the atypical AD syndromic spectrum. Photography is welcome, and I have no disclosures. Okay, so what are atypical syndromes? So these refer to clinical variants in which non-amnestic syndromes dominate early in the disease course. And these variants tend to be enriched in early onset cases. So we have a visuospatial predominant syndrome that we refer to as posterior cortical atrophy, or PCA. We have a language predominant syndrome that we call logopenic variant of primary progressive aphasia, or LVPPA. And then a dysexecutive predominant syndrome that we refer to simply as dysexecutive AD. So of course there are a spectrum of other variants of the AD kind of course of symptoms, but I will refer to these three in particular for the purposes of this talk. So these variants tend to arise from initial selective vulnerability of non-medial temporal cortical networks, though there are also common networks of vulnerability, for example, the default mode network, that are common across the syndromic spectrum of Alzheimer's. So we need assessments that can concisely characterize the profiles of performance across the entire syndromic spectrum in order to inform individualized treatment and outcomes monitoring. So we all know that typical older onset AD is characterized primarily by memory impairment, and early onset cases are more often considered to be non-amnestic. So this might be an oversimplified characterization of syndromes based on the initial domain of impairment. Indeed, memory decline is reported by patients with atypical phenotypes of AD and their families even early in the course of illness. For example, Shear and colleagues just earlier this year reported that 62% of their PCA cohort reported memory problems. Now, the clinicians among us know that when patients come in talking about memory problems, it's imperative that we ask further questions in order to disentangle the actual memory storage loss from other perceptual vulnerabilities that could be impacting their experience of memory problems in day-to-day life. Towards this end, it's important to realize that memory is not one monolithic entity. Memory is itself a multi-domain cognitive construct. So first, if we need to learn information, this encoding phase relies on working memory and material-specific perceptual processing. This relies on multiple different cortical networks. We then need to store the information that we learn, and this retention really localizes to the medial temporal lobes. And last, we need to recall the new information that we learned. And this relies on goal-directed retrieval skills, largely relying on frontoparietal and lateral parietal cortices. Ultimately, we think that memory testing, because of all of these features, can be a useful lens through which we can examine different cognitive processes across the atypical phenotypic spectrum of AD. There's in fact been increasing evidence of memory deficits in PCA, LVPPA, and dysexecutive phenotypes at early stages of illness. For example, Ahmed and colleagues in 2016 show that PCA and early onset AD showed comparable memory impairment on a screening tool at all three stages of memory, encoding, storage, and recall, even at initial clinical presentation. We've also shown verbal encoding and retrieval deficits that were strongly related to verbal working memory deficits, while storage or recognition discriminability was not. And then we built on this to show that PCA and LVPPA, shown together in the red line here, benefit from repeated presentation of verbal information, while the amnestic syndrome, shown in blue, showed a flat learning curve, so they did not benefit. Taking all this together, we're realizing now how we measure memory matters in order to figure out what the actual cognitive profile of these phenotypic syndromes are. So now I'll introduce this new concept of object location binding. Performance on object location binding tests is disproportionately impaired in early stages of AD, reflecting early hippocampal and posterior parietal network dysfunction. Now, these deficits are thought to be more sensitive than item or wordless memory tests for detecting the earliest stages in preclinical and prodromal AD. Indeed, binding impairments do differ across AD phenotypes. So the typical amnestic AD syndrome shows global binding deficits, while PCA may show relatively preserved object memory, but impaired spatial binding. And logopenic PPA may exhibit broad encoding and verbal working memory deficits. So our goal for this study was to develop a novel associative memory paradigm testing object location binding in order to tease this apart and evaluate if this measure differentiates between atypical syndromes. In the end, the better we can characterize these shared and dissociable patterns of impairment across the atypical spectrum, the better we can prognosticate and measure personalized response to treatments. So now introducing this object location memory test. As I mentioned, this is an associative memory paradigm, and we designed this to facilitate active and deep encoding to really support the learning process, so then we can infer actual storage and recall deficits. So during the learning phase, we presented participants with a picture and asked them to name it. In this way, we paired a visual cue with an auditory cue, again, emphasizing that deep encoding. They were shown 16 pictures in total, and this was repeated 3 times. Each object was also associated with a particular spatial location on the screen. Memory for objects was tested with forced choice recognition, and this was on purpose to minimize reliance on lexical retrieval. So for example, instead of asking participants to name the objects that they learned, we just asked them, is this old or new? And then we tested their memory for these objects after a 3-minute delay and a 30-minute delay. This 3-minute recognition test was thought to measure rapid forgetting, while the 30-minute delay was more of a measure of storage loss over time. How do I go back? Okay. Now, memory for locations was also tested at 3 and 30-minute delays. In this way, we could really dissociate object memory from location memory. So this is an example of what a participant might see. So we show them a picture of a bear in box 1, picture of a cup in box 2, picture of a candle in box 3, and a lobster in box 4. And this repeats 3 times. And after a 3 and 30-minute delay, we'll ask them, is this picture old or new? If they indicate that it's old, we'll ask them, which box did you see it in? And hopefully all of you know the correct answer for that one, even at this hour. So we studied this task in a total of 49 participants, 23 of whom met the criteria for posterior cortical atrophy, 10 met the criteria for LVPPA, and the remaining 16 were best characterized as having an amnestic dysexecutive syndrome. This amnestic dysexecutive group were younger than the PPA group, but matched with the PCA group. Otherwise, all 3 syndromic groups were matched on sex, education, global clinical severity measured by the Clinical Dementia Rating Scale, and global cognition measured by the screening tool, the MoCA. So here's our first major finding. We found impaired object encoding across the atypical syndromes. So compared to a group of 29 control participants, all 3 groups showed impaired encoding. Notably, the amnestic dysexecutive group was relatively less impaired compared to the PCA and LVPPA groups in learning objects. On the y-axis, I'm showing you the z-scores for the patient scores compared to the control participants. So you can see the PCA in orange and the logopenic PPA group in the green are more impaired than the amnestic dysexecutive group in blue. And again, this is total object encoding across all 3 trials. Now, when we break performance down by each trial, we see that each group also benefited from repetition of those objects. So although the PCA and LVPPA groups started out lower on trial 1, they benefited significantly from repetition, and by trial 3, shown in the green, you can see that they're fairly comparable to the amnestic dysexecutive group. When we looked at memory for objects after a short delay, after just 3 minutes, we found significantly impaired performance in the amnestic dysexecutive group. So despite showing relatively stronger encoding, we found that this group, the amnestic dysexecutive group, had the greatest immediate storage loss. In contrast, PCA and LVPPA groups retained object memory after a 3-minute delay. The story changes after a longer delay. So where we might think that PCA and LVPPA had intact storage if we test only at the 3-minute mark, when we wait and test them again after a 30-minute delay, we now show that all groups show comparable storage loss over time. So in sum, the amnestic dysexecutive group lost object memory early, shown in the blue bars, but all groups ultimately ended up with storage loss for objects at the 30-minute mark, shown in orange. Now, what about location memory? So we found actually that location encoding was only mildly impaired across each syndrome, and were comparably so. However, when we measured location memory over time, we found that this declines in the amnestic dysexecutive and PCA groups in particular. So if you focus on the first cluster of bars, you can see that by 3 minutes, the amnestic dysexecutive group showed markedly reduced memory for locations, which was consistent after a 30-minute delay. The PCA group, interestingly, had a stepwise decline in location memory. So while encoding was relatively spared, we see that they lose this memory for location binding at 3 minutes and further at 30 minutes. In contrast to these two groups, location encoding and retention were intact in LVPPA. So to conclude, we saw initial encoding of new object information that was impaired across the board. This was more prominently impaired in PCA and LVPPA, more so than in early onset amnestic dysexecutive patients. We think there are contributing perceptual vulnerabilities as well as limited working memory buffer that could have contributed to the performance on encoding that we saw in PCA and LVPPA in particular. We also observed that encoding improves with repetition across these phenotypes, and this is in contrast with a flat learning curve observed in late onset typical amnestic AD. This has big relevance for cognitive skills training and functional support. As we learn how to build compensatory programs to teach these patients how to improve their independence and functional activities in daily life, we really need to remember that we need repetition of instruction and to be able to rely on external compensatory strategies in order to store these new skills that we teach them over time. We saw object memory storage loss that was observed earlier and most prominently in the amnestic dysexecutive group, but still present after a longer time period in PCA and LVPPA, even in the absence of lexical retrieval demands. Location memory storage loss was observed only in the amnestic dysexecutive and the PCA groups, but not in LVPPA. And we wonder if the mechanisms of this impaired performance may be different. Are there differential roles of the medial temporal lobes versus the posterior parietal cortices in showing this location binding loss differently in these syndromes? So this novel object location memory test can capture overlapping and dissociable patterns of memory deficits across these 3 atypical AD variants. And this is consistent with many patient reports of memory deficits in day-to-day life. This pattern of deficits likely reflect different contributing domains of impairment beyond memory storage, specifically executive control, working memory, and perceptual processing deficits. Ultimately, understanding these contributions to memory can inform targeted cognitive skills training. So there are 2 major questions that I'll end with in terms of future directions for moving this field forward. The first is, how do these memory profiles track with clinical severity over time? Now, data collection is ongoing as bigger sample sizes are needed. But in order to address this question properly, we also need to think about, as a field, how do we measure clinical severity in atypical forms of AD? The traditional methods designed for typical amnestic AD, such as the Clinical Dementia Rating Scale, just may not be sufficient. The other major question we want to ask is, how do these profiles track with longitudinal tau accumulation? And hopefully, in the not too distant future, how do they track with tau clearance? In the end, a biologically validated tool useful for measuring cognitive decline across the entire phenotypic spectrum of AD will be needed as a candidate outcome measure in designing clinical trials. So with that, I want to first acknowledge the time and effort of our research participants, without whom this work would not be possible. I would also like to thank my colleagues at the Massachusetts General Hospital Frontotemporal Disorders Unit, Yuta Katsumi and Brad Dickerson, as well as the hard work of our clinical research coordinators, Canella Basilian, Gent Salaj, and Sophia Cheer, for their hard work in recruiting and testing these participants. I'd like to thank the funding sources and thank you all for your attention.
Thank you, Dr. Pucha, for an amazing talk. Before we move on with our next speaker, I quickly want to introduce us. We are Dr. Kier Young, who's the chair of the Atypical AD PIA, and my name is Baila Boon. I am the vice chair of the Atypical AD PIA. Our next speaker is Dr. Valgas, and she will talk to us about sleep disruption in early onset Alzheimer's disease.
Good morning, everybody. I'm very happy to be here today to explain a little bit our work revolving around sleep disruption and neuromodulatory subcortical systems in early onset Alzheimer's. As many of you know, the early onset Alzheimer's disease definition is defined by the fact that the age of onset of the individuals with AD starts before 65 in opposition to the late onset Alzheimer's. This 65-year-old cutoff might seem arbitrary, but in fact is useful because this group of individuals have specific features. For instance, there is a predominance of the atypical or so-called non-amnestic variants of AD. And they also have more aggressive disease curves even between the first years of progression, not only globally, but also specifically within the non-amnestic domains. But besides the cognitive differences that these two groups may show, we also, sorry, it was a change in the slide. It also shows that several studies that we have performed show that there are differences also in regards to the neuropsychiatric symptoms and how they manifest in these two different groups. So this recent study performed at UCSF showed that the early onset Alzheimer's disease were having higher severity of neuropsychiatric symptoms, not only at the diagnosis, but also longitudinally, and specifically in specific domains such as the depression, anxiety, or nighttime behaviors. Furthermore, another study using polysomnography in a group of early onset Alzheimer's disease showed there are differences in a sleep architecture. That means that the sleep stages were organized a little bit different, being that the amnestic AD were having a lower percentage of slow wave sleep. This is the N3 of the non-REM sleep. And the non-amnestics AD were showing a decreased proportion of REM sleep. And overall, we see that there is this wide clinical heterogeneity, not only in cognition, but in neuropsychiatric symptoms. And in the last decades, we have gained a lot of more understanding in why this heterogeneity in the manifestations is happening. But especially, we did a great job in understanding why patients may manifest with atypical or typical cognitive deficits. We know now that the distribution of tau within the cortex is really relevant. So some patient that has a lot of tau located in the hippocampus, that would lead to an amnestic AD form. If this tau is located in occipital, probably this patient would develop a PCA. And we also understand that the fact that they have a more aggressive disease score, that they decline faster, it's probably related to the higher tau burden that within the cortex that these patients also show. But on the other hand, we don't understand very well why there are differences in the manifestation of the neuropsychiatric symptoms or sleep symptoms across the different AD variants or even the early and late onset Alzheimer's disease. We try to disentangle this in the last recent years. And before I start to showing you some results or the work that we are doing, I wanted to stop for 1 minute to introduce you the neuromodulatory subcortical systems, which are a part of the brain really relevant in regard to these syndromes. The neuromodulatory subcortical systems are a group of very, very tiny nuclei that are interconnected within the brainstem and basal forebrain, and they have projections through the brain, and they release different neurotransmitters. Each of the nuclei have a specific neurotransmitter. For instance, the nucleus basalis of Meynert is releasing acetylcholine, the substantia nigra, the dopamine, there is orexin, histamine in the hypothalamus, and there is one special that has gathered more attention in the last years, that is the locus ceruleus, which is the main source of noradrenaline in the brain. The relevance of the locus ceruleus that gathered a lot of attention on the last years is due to the fact that it was recognized by the Braak staging as being one of the first areas of the brain of being affected by tau pathology, even before tau pathology reaches the hippocampus. So there is a lot of interest in this nuclei, but there is a lot of research going on and trying to investigate other nuclei and we have growing evidence that other nuclei are also affected at the very, very early preclinical stages. And not only the fact that LC is affected so early, but also the fact that there are a lot of associations between these nuclei, how it degenerates, and the degree of neuropsychiatric and sleep symptoms. We have postmortem correlations with clinical data showing that the odds of developing anxiety, depression, or sleep disturbances is higher when the tau is restricted to the locus ceruleus and barely has reached the hippocampus. And this has been validated also by other human in vivo studies, not only with the neuropsychiatric symptoms, but with polysomnography and actigraphy studies correlating the LC integrity with the sleep disruption. So our first question, putting all of this in mind on the last years, was try to understand whether the locus ceruleus could have a role in the fact that the early onset Alzheimer's disease were having an increased severity of the neuropsychiatric symptoms. And we did this study published last year in which we found indeed that the early onset Alzheimer's were having lower locus ceruleus integrity, which is a specific measure for the locus ceruleus in the MRI. Compared to the late onset Alzheimer's disease. And this was happening independently of the disease stage. And it correlated with the neuropsychiatric symptoms. That was good news because we help us to understand a little bit, but still there are a lot of knowledge gaps. For instance, there are lack of cohorts studying sleep in early and late onset Alzheimer's disease by using sleep objective measurements. And we don't know much about other nuclei besides the locus ceruleus. So we try to partially address that by comparing sleep wave patterns in a cohort of early and late onset Alzheimer's, and also comparing the volumes of the NSS nuclei. In order to do this, we included a sample of 113 participants, all of them in early stages of the disease, MCI, mild dementia, and confirmed by biomarkers at the Hospital Clínic de Barcelona. All of them underwent an MRI scan, including the neuromelanin-sensitive MRI, to be able to measure the locus ceruleus integrity and volume, and also segmenting other areas using other protocols and get the volumes of the hypothalamus, substantia nigra, dorsal raphe, basal forebrain, and other limbic areas. A subsample of participants also underwent actigraphy monitoring during 2 weeks. The results in regards to demographic and clinical data show the obvious difference in age and age of onset as expected, but we didn't find differences in terms of disease stage. Most of them were MCI stages. Cognition was similar across groups, and also the amnestic phenotype was predominant. There were not major differences in the prescribed medications either. The actigraphy results show that the early onset Alzheimer's disease were spending less time in bed. They were having less time sleeping during this time in bed. And also they were less time immobile, which is clearly correlated to the fact, to the sleep period. So they have less sleep in general, around a bit more than half an hour less than the late onset counterparts. And derived from these results, this suggests that there is a decrease at the sleep drive in early onset Alzheimer's disease compared to late onset. Although these are preliminary results because we have a lower sample of early onset, we are trying to increase them. But this is, at this point, what this suggests. And this is in line of the qualitative assessment as well. Gerard Maya, who is a sleep neurologist collaborating with our unit, is evaluating actigraphies visually and trying to detect abnormal patterns that might be missed by the quantification. And he found that the abnormal patterns were more frequently present in early onset AD, especially because of a delayed phase phenotype and also sleep deprivation. Then I'm going to show you the results of the volumes of the neuromodulatory subcortical systems, and I'm going to start with the basal forebrain. In this case, we found that the basal forebrain was more preserved in early onset in comparison to late onset Alzheimer's disease. At first, this was not surprising because we also found in another cohorts that hippocampus, for instance, was also more preserved in early onset AD. So we think that the late onset have this added age effect, which in general the brain is more atrophied. However, this result is somehow divergent with other studies that have been performed neuropathologically. I'm showing here the results of a great study performed at Mayo Clinic by Melissa Murray's lab that they also show that the nucleus basalis of Meynert was degenerated differently across different AD subtypes, which is in line of our hypothesis. However, they found that the age of onset were affecting negatively to the neurofibrillary tangular accumulation and degeneration. This might be a bit conflicting with our studies, but probably they are not comparable. We're measuring different outcomes, and in our end, the basal forebrain that we are measuring includes the nucleus basalis of Meynert, but it's not the only nuclei that might be involved. On the other hand, a similar result was found with the hypothalamus, including the lexigenic histaminergic nuclei, that was more preserved in early onset compared to late onset. We didn't find any statistically significant differences with the dorsal raphe or other nuclei, such as the substantia nigra or other limbic structures that were added as a way of control. We validated and replicated the fact that the early onset AD were having lower locus coeruleus integrity, but also lower locus coeruleus volume. So all the measures are in the same direction. And this suggests that there is a selective vulnerability of these nuclei, especially in early onset Alzheimer's disease. And happily, this week we presented in a poster of a work also performed at the Mayo Clinic. And it seems that neuropathologically, the trends are going to the same direction. So I'm happy that we are maybe not agree a lot in terms of the nuclei basalis of Meynert, but we are confluent in the locus coeruleus. So as a conclusion, I would like to extract from this study that the sleep patterns are different in early and late onset AD, and so are in the neuropsychiatric symptoms in general. From this study, the data that is preliminary suggests that the sleep drive is more decreased in early onset Alzheimer's in comparison to the late onset AD, but there is still a lot of work that we need to do to make this, to validate this. And in addition, we have prior studies showing that not only the age of onset, but also the AD phenotype may vary the sleep architecture importantly. The locus coeruleus is particularly affected in early onset AD, suggesting this special selective vulnerability. And this may have an impact in all of the symptoms, neuropsychiatric and sleep symptoms that we were mentioning. Definitely, we need further studies to better understand the relationships between and the effect of age of onset, the clinical phenotypes, and the degeneration of this nuclei to try to disentangle the clinical heterogeneity of Alzheimer's disease as a whole. And we need larger sample sizes, but it's always challenging because early onset Alzheimer's atypical, they are difficult to recruit, and especially when you are performing sleep studies. Also, we need better imaging to segment properly NSS. That's very challenging because they are very tiny nuclei. And I think that it's key to have this neuropathological and also in vivo data contributing together to validate any changes. I would like to thank everybody at the Hospital Clinic de Barcelona, the Alzheimer's unit, the sleep unit, MRI platform, and also my beloved mentor, the amazing Leah Greenberg. And thank you, everybody, for listening.
Next, we have Dr. Rosalina Mahanti from the Karolinska Institute.
Thank you very much for including me in this super interesting session, and thank you everyone for being here. I'm going to be talking about imaging heterogeneity in Alzheimer's disease, and I have nothing to disclose. I would like to contextualize the talk for today in the diagnostic criteria for Alzheimer's disease, which lays out the core biomarkers that are relevant to the disease. And in particular, I will be focusing on the imaging biomarkers. So I would like to quickly walk through the different subtyping or studies that have looked at heterogeneity from the imaging perspective by going through these core biomarkers. Starting with core 1 biomarker, A, or which can be imaged by amyloid PET. In the past couple of years, we've seen many data-driven approaches that have been able to identify and capture heterogeneity in amyloid patterns in the brain. This study in particular identifies 3 different subtypes based on the SUSTAIN model. And there have been other studies that have also identified a more cortical versus a more subcortical pattern of amyloid pattern in the brain. Looking at the core 2 biomarkers representing tau pathology in the brain, which can be imaged using tau PET imaging. There have been several seminal studies, starting with neuropath and then translated back to neuroimaging data with tau PET that have identified different subtypes or different patterns of tau pathology in the brain, predominantly looking at corticolimbic subtypes of tau pathology. Then coming to the non-specific markers, looking at neurodegeneration in particular. If we think about structural MRI in particular, very similar to tau PET data, we've seen corticolimbic atrophy patterns representing atrophy subtypes in several of the previous studies. And when talking about neurodegeneration, we also have FDG PET imaging modality. And in the recent years, again, studies have now identified subtypes of FDG PET that reflect hypometabolism patterns in the brain, yet again reflecting corticolimbic patterns. Altogether, several of these subtypes that have been identified, particularly in tau PET and MRI and FDG PET modalities, have been or can be summarized in this meta-analysis and conceptual framework that looks at subtypes based on imaging by looking at 2 different dimensions. Along the horizontal is the typicality dimension, which determines how typical or atypical someone is in terms of their neurodegeneration or pathology. And individuals can be differentiated based on whether they exhibit a hippocampal sparing pattern versus a limbic predominant pattern. Along the vertical, we have the severity dimension, which captures the overall burden of the pathology or overall neurodegeneration. And it separates, for example, a typical pattern from a minimal atrophy or a minimal tau pattern. All these different subtypes or patterns are associated with unique features such as the demographics, the sex, risk and protective factors, co-pathologies. So with that framework in mind, I want to discuss 3 main points in today's talk. So we've been trying to look at, in the first place, given that we can identify subtypes based on different imaging modalities, how well do subtypes translate across modalities is one of the research questions of interest for us. To do this, we have looked at the typicality and the severity dimensions in terms of tau PET data, and we have looked at the 4 subtypes mentioned earlier, namely typical Alzheimer's disease, cortical predominant, limbic predominant, minimal tau subtypes. And using the baseline tau PET data, we then wanted to understand how they track if we were to look at longitudinal atrophy based on structural MRI data. So we looked at retrospective as well as prospective structural MRI to look at the atrophy. At baseline, when we look at tau PET data, we were able to identify a typical Alzheimer tau pattern, a limbic predominant, a cortical predominant, and a minimal tau pattern. And when we look at the longitudinal atrophy, these are the topographies that we see. We looked at how closely atrophy tracks the baseline tau pattern. And what we observed, and we quantified this overlap in terms of dice overlap, and we observed that the dice overlap increases over time when we look at typical and limbic predominant tau patterns in particular, whereas when we look at cortical predominant and minimal tau patterns, the atrophy patterns do not identically mimic the tau PET pattern, suggesting that there is a differential association between tau PET pattern and atrophy. Along the same vein, Sophia Wheatley, who is a PhD student in the lab, has been looking at heterogeneity in FDG PET as well as structural MRI. Given that they both are measures of neurodegeneration, we have quantified the subtypes in a data-driven way using a clustering approach and identified corticolimbic subtypes based on hypometabolism in FDG PET and atrophy patterns in MRI. Yet again, we see that when we compare the FDG PET subtypes to the MRI-based subtypes, at the individual level, there is a lot of mismatch or discordance. Further, what we wanted to do is we wanted to apply a number of different subtyping methods on the same set of individuals to see how the different subtyping methods compare. And to do this, we've looked at hypothesis-driven methods, data-driven methods, some based on automated whole brain volumes or measures, and some based on visual ratings that are more clinically relevant. What we see is that when we compare along the horizontal and the vertical are the different subtyping methods, the Cohen's-kappa values are relatively low, suggesting that there is a lot of disagreement between different subtyping methods. At the individual level, if we zoom in, along the columns are 4 different subtyping methods, and they classify each individual which is listed along the rows, and we see that not every individual is consistently allocated to be in the same subtype by the different methods. These findings suggest that subtyping in different imaging modalities do not translate, and they are not interchangeable, even if we are looking at similar biological processes such as neurodegeneration. And importantly, we also need to recognize that subtyping methods do not always agree with each other, and we need to better harmonize them for translation for trials. The second aspect that I would like to discuss is what is the contribution of co-pathologies. We wanted to test whether co-pathologies can explain some of these discordances that we see in subtyping across imaging modalities. Again, study led by Sophia Wheatley. She looked at FDG PET-based subtypes and MRI-based subtypes in the same set of individuals, identifying corticolimbic patterns. And when we compare the two modalities, we see a relatively low agreement between the two modalities. So we wanted to assess the role of co-pathologies based on autopsy data. So we looked at cerebral amyloid angiopathy, arteriolosclerosis, alpha-synuclein, TDP-43, and hippocampal sclerosis. The bar plots indicate each individual, and we see FDG and MRI PET subtypes compared on an individual level basis. The differences we see are when we stratify by the AD neuropathologic change. For no or low AD neuropathologic change, we see that there is no disagreement between FDG PET and MRI subtypes. However, for intermediate or high AD neuropathologic change is where we see that FDG and MRI subtypes disagree, which may indicate that we need the interaction of AD pathology with co-pathologies to induce this type of discordance in neurodegenerative subtypes. Further, she has also looked at different co-pathologies at a regional level in neocortical as well as medial temporal regions in the brain. And we see that when we look at FDG PET-based typicality and severity measures compared to MRI-based typicality and severity measures, they differentially associate with co-pathologies, in particular limbic, cerebrovascular, and alpha-synuclein co-pathologies. Going back to the diagnostic criteria, we now also recognize V, or the vascular pathology, as one of the key or one of the relevant pathologies in the disease process. However, vascular pathology is quite heterogeneous in nature and can manifest in many different forms. So to address this, we wanted to take an antemortem imaging and postmortem autopsy validation approach. So we started by looking at 8 different markers of vascular pathology based on autopsy data in ADNI dataset. And we applied a data reduction approach, and we identified the 2 most important variables, CAA being one of them, and arteriolosclerosis being the other. We then correlates of each of these 2 cerebrovascular markers in terms of correlates at the neuroimaging level, the cognitive data, and neuropathology data. So we see that arteriolosclerosis and CAA are differentially associated with markers of neuroimaging, cognition, and neuropathology, with CAA being a relatively more AD-specific marker, whereas arteriolosclerosis is a relatively non-AD-specific marker. Specifically, the interactions that were significant were individuals with greater white matter lesion burden in the brain and greater hippocampal atrophy showed greater burden or greater severity of arteriolosclerosis. When we look at CAA, we observed that diffusion matter changes in posterior and cerebellar tracts in the brain were associated with greater severity of CAA, and greater CAA and having a greater AD neuropathologic change was also associated with greater cortical atrophy as well as poorer memory scores. Altogether, these few studies suggest that we need to account for copathologies when we look at the subtype characterization based on imaging, and we need to account for not only global level of these copathologies, but also the regional level of these pathologies, if available. An important reflection from these studies has also been that copathologies may be heterogeneous in nature within themselves and can then differentially be associated with the different imaging subtypes. And I finally want to discuss the role of accounting for imaging-based heterogeneity or subtypes in trials. And I want to highlight this one study done by a previous postdoc, jointly led by a current postdoc in the lab as well, where we looked at the hippocampus study from France, which recruited individuals with prodromal AD, and individuals were randomized into either the donepezil arm or the placebo arm. And we looked at MRI data at baseline before the treatment and the follow-up after the treatment. We subtyped the individuals based on atrophy measures, looking at typicality and severity at baseline. And we assessed the efficacy outcomes at the biological level by looking at % change in hippocampal volume, lateral ventricle volume, gray matter volume, and AD signature thickness. We found that, in particular, individuals in the donepezil group who were hippocampal sparing atrophy patterns were likely to show lesser ventricular expansion and lesser thinning in the AD signature areas. Similarly, along the severity dimension, we observed that individuals in the donepezil group who show minimal atrophy in particular show lesser thinning in the AD signature area. Altogether, suggesting that we could potentially use the imaging-based measures of heterogeneity, such as typicality and severity, to also track treatment response, and that subtypes can actually reflect differential response to treatment. And this is only a proof of concept for a symptomatic treatment, but it would be interesting to see how it generalizes to the upcoming treatments in the future. And I just want to close by saying that we really need to develop such subtype-specific measures, whether clinical subtypes or biological subtypes, to really move towards precision medicine. With that, I want to thank all the members of the lab at Karolinska Institute who've contributed to these studies, all the funding sources that have supported these studies, and thank you for your attention.
Thank you, Dr. Mohanty. We continue with our last speaker of today, that is Dr. Leah Grimberg. She will talk about cellular vulnerability and concomitant pathology in atypical Alzheimer's disease.
Hi, everybody. It's very good to be here, and thank you for the very active Atypical EDP for putting this together and inviting me. So in my lab, we work with post-mortem tissue trying to understand heterogeneity of Alzheimer's disease, and I would love to share some of these studies with you today. So just to start from the beginning, so we are everybody on the same page when, at least with the current the current day and era, when we think about Alzheimer's disease, we are talking about a pathological process that we have accumulation of plaques and tangles in the brain, follow a topographical spread, and which is a little bit different in amyloid, starting in neocortex and then progressing to allocortex, going to subcortical area, cerebellum, and the tau spread, which starts in the neuromodulatory subcortical system, as Dr. Falgas was showing, entorhinal cortex, and then it follows to the neocortex. So if they have plaques and tangles, usually following this progress, this is Alzheimer's disease, and this is the definition I will use right now. Now, by using this definition, I think we all can recognize that Alzheimer's disease is very heterogeneous, and there are some questions if it's really a disease, so you have one cause, one pathological progression, and then the outcome, or if actually what we are looking is a pathogenic convergence, maybe different kind of triggers that can lead to same pathological characteristics, like liver cirrhosis, for instance, could be viral, could be alcoholic, could be other things, but they kind of look around the same under the microscope. So this is some of the hypotheses we are trying to work with in the lab. And for this, the idea of atypicality in Alzheimer's disease come very handy. So atypicality in the sense that most of the cases with this Alzheimer's disease pathology will have a evolving amnestic syndrome from the beginning. But there are some cases, when we look at the microscope, they are the same plaques, the same tangles, they look really alike, but they express with different clinical syndromes like language and logopenic variant PPA or behavior, visual, spatial, and so forth. So by looking into this, the question comes, what is the neuropathological substract of this atypical Alzheimer's disease presentation? And also, if we can use these AD variants, I will call these variants because there are several, even in the atypical, as an approach to unveil this concept of selective vulnerability in Alzheimer's disease. What we can learn that can help us to protect our brain or our cells to develop and to spread this disease. So we talk a lot about heterogeneity and co-pathology, and very elegant presentation before mine. Of course, co-pathology is the rule in the aging brain. It can modify the clinical symptoms. But the question we asked was a little bit different. If we take people who come to autopsy and they have a primary diagnosis of Alzheimer's disease, plaques and tangles, and then we look at co-pathology, will this co-pathology be able to define the clinical presentation as we define these AD subtypes? Would it meet criteria for logopenic variant PPA or PCA or the behavioral frontal? And we try to study this with this cohort at UCSF. And for this study, we have about 150 brains, and 60 of them were in the atypical category. So when we compare the typical and the atypical, especially excluding people who were late onset Alzheimer's disease, so we didn't have really the issue of the age, we noticed that the atypical cases, they were younger, they were more likely to be female, they had smaller brains, they were less likely to have a APOE4, but most important for this specific conversation now, they have less number of co-pathologies than the typical case. And when we compare all the different modalities of co-pathology like late or AGD or Lewy body disease, we didn't find difference in typical and atypical. Actually, we found one difference, that actually the typical ones were more likely to have vascular pathology in the brain. So with this study, asking these specific questions, if co-pathology could define one of these typical variants, the response was no. But despite this, these atypical cases had accelerated decline compared to the typical cases, which bring us again to these questions of the selective vulnerability. So why this is happening? So given that co-pathology for this specific question is unlikely to explain the atypical presentation, we went to the next step. And one of these very important one is to understand the regional tau burden. Because we know from several studies when we look at amyloid and tau, tau is a better predictor of cognitive decline. And again, they come from different directions in the brain, so it's important to differentiate both. And we were very inspired by the work that Melissa Murray has been doing since the late 2010s, showing that with neuropathology, we could, for tau, we could distinguish different neuropathological subtypes of Alzheimer's disease. So we try to take this idea, in our cohort at UCSF, which is a little bit different from Dr. Murray cohort from before, because these cases are deeply phenotyped, especially we know well if they are atypical or not, because they were followed up in the clinic. So for this particular study, we had 94 cases, and we used thioflavin S, which is a dye that we can use to detect mature tangles, and we look at the density of these tangles in different areas of the brain. And we did a little bit, it's not going forward. We did a little bit of a different process that Melissa did. We did unbiased clustering. And then we plot the density of these tangles in these different cases based on the clinical phenotype. Again, cases that meet criteria for cortical basal syndrome or logopenic variant PPA, the amnestic/dysexecutive syndrome, and this graph illustrates what we found. So, for instance, let's go here to the cortical basal syndrome, and this area here that's darker means more tangles. This is the premotor area, right, where it's really linked to CBS, which is the syndrome here. And if you look at this compared to all the other subtypes, we did find more tangles here. Now let's look at logopenic variant PPA. If you look here at this area, the posterior superior temporal gyrus, which is responsible for the symptoms, we saw in this case that it was darker than the other ones. So with this kind of relatively simple overlay, we could illustrate well that it seems, yes, as PET studies have been suggested, and now I saw some more neuropathological studies are coming out. We have important regional difference in tau density that can correspond to some of these different clinical presentations in Alzheimer's disease. So I got very excited, and we said, okay, now we have a model that we can probably use to try to understand this heterogeneity in Alzheimer's disease on a cellular level, on a tissue level, and indeed we joined forces together with Leeds and Dino Getty in Indiana University, Melissa Murray at Mayo, and I was at UCSF at that time. We got the R01 to try to study this. And we did a lot of things. I have shown some of this before, but today I want to show something, a little bit new concept that we are trying to do to understand these differences. And I want to go back to the spread, the stereotypical spread of Alzheimer's disease, and call attention specifically for where tau, how progress. So we have these very early stages in the neuromodulatory subcortical system. It's not only that this is driven by tau, but these are areas that amyloid barely touch. So if we want to look at tau and how tau makes different, heterogeneity in Alzheimer's disease, we felt it could be a good area to look at. And it's something that my lab has been studying for many years, so we felt we have the tools to do it. And when we studied the neuromodulatory subcortical system, as Nels Falgas explained very well, this is an area that starts to be affected before memory changes of other changes. So we need to have some clinical outcome that we can measure. And all that kind of correlate with these early stages of degeneration. Sleep is one that we can measure. We can do EEG, we can do actigraphy. So this is why we try to combine these sleep measures with changes in this nuclei when we are studying them. So Nels explained a little bit about sleep, but for those who are not very familiar, when we think on sleep, is a cycle of 24 hours, right? So we have nuclei in the brain that control our awake. They make us be awake, alert, and there are some other areas, they are completely different, that control our sleep. And sleep has two major elements I put here in green. This is the no REM sleep. And the no REM sleep is controlling the brain in different areas that the yellow, which is the REM sleep. And finally, we have this red circle here. This is the suprachiasmatic nucleus, master clock circadian, right? So when I will be talking about the studies right now, I will talk about awake, different area of the brain that no REM sleep, then REM sleep, then circadian. And this will be important to understand some of the results. And we like these areas I showed before because they are affected early in Alzheimer's disease, but also in a different chronological timing, with the awake nuclei coming first and then they sleep. So again, a kind of approach to understand heterogeneity. We had some studies before in which we examined these areas and we showed that there is a direct correlation between changes in neurons there with clinical parameters of sleep measured by, for instance, polysomnography. So again, it gave us more confidence that what we'll be studying there, we can try to understand if there is a clinical correlation to understand heterogeneity. Now, how does it come to Alzheimer's disease and atypical Alzheimer's disease? Now, Falgas presented before her studies showing that when she compared the pattern of polysomnography with people that have typical Alzheimer's disease amnestic with those that were atypical, and the atypical group were really logopenic variant PPA and PCA, she noticed that the atypical ones, they have much more compromised REM sleep than the typical ones. And as opposed to the typical ones that have no REM sleep, we talk a lot about no REM sleep in Alzheimer's disease, right, the lymphatic system, etc. But the atypical cases, they didn't have no REM sleep problem. So I said, well, that's great. Let's try to follow up and see what is going on in these neurons that control these symptoms in the brain, in the tissue level. So the first one we look at, and, well, let's go here. The first one that we look at, it's a population of neurons in the anterior hypothalamus that's called intermediate nucleus of the hypothalamus. And for those who study sleep in rodents, you might recognize this as the VLPO. The VLPO is the main driver on no REM sleep in the brain. So our hypothesis was that if it's true that no people with atypical Alzheimer's disease had preserved no REM sleep compared to typical, if we would just count neurons, and these neurons are galanin neurons, this is the GAL, we will see that probably we will find less loss in the atypical than the typical. And this is a work that Shima Hastegar in my lab did with stereologists, time consuming, but this is exactly what she found. We found that in atypical Alzheimer's disease that come to death, so they are late stage, they still have the double of the number of neurons in these areas than in the amnestic. So, you know, maybe this is a one-off, maybe it's because atypical Alzheimer's disease, they die early, so they didn't have time to lose neurons, so we had to look in another area, right? And this is exactly what we did. And this time, we look at an area that controls REM sleep. So REM sleep is the one that is worse in atypical Alzheimer's disease. And the type of nucleus that we look at is called melanin concentrating hormone. And this is a very important neuronal population located in the lateral hypothalamic area, very close to the rexinergic neurons, one of the main drivers of REM sleep. So our hypothesis here, it will be the difference that in atypical, we will have more neuronal loss than the typical, if you were right. And this is exactly what we found. So in this graph, the first column are controls, the second are amnestic, and the third are typical. So we only found the statistical significant changes in the atypical ones. There is a chance that where we increase the sample size, we are doing this right now, we don't have many amnestics, it will change a little bit, but it will be, I will be very surprised if we will see that the amnestic will be similar or than the atypical ones. Especially because we have a good number of atypical, and again, this is stereology, very unbiased counting. So again, we are excited for this, maybe this is not just one-off, so we move forward, and we did some RNA sequencing to try to understand if the pathways that we see dysregulated in amnestic AD is similar or different from non-amnestic. Because again, our goal here is to use these systems to understand heterogeneity. And kind of for our, it was surprising, but a good surprise. Indeed, we found that the dysregulated pathways are different. in which the no amnestic it's much more metabolic, the amnestic is more inflammatory, which go in line with some other comparisons we are doing with typical and atypical in other areas. Now, finally, we decided just to look at again, a different area, right? I mean, this is post-mortem, it's late stages, it's cross-sectional. We cannot just draw conclusions by looking at one area. So we finally went to the SCN, the suprachiasmatic nucleus circadian. So Christine Walsh and Tom Nylam at UCSF, they have been following a living cohort, and they detected that the atypical Alzheimer's disease, they will have more circadian dysfunction than the atypical one. The atypical one is much more similar to controls than the typical. So again, the hypothesis here is that we'll find more changes in the SCN in the typical ones than in the atypical. So we counted, great judge who just presented before me and Gu-Woon Song, they did these countings, and this is exactly what they found. So here in the first graph, this is control number of neurons, and here's what happened to amnestic, and here is where the atypical ones. So again, it goes in line now for the third time, and hope third time is a charm with our hypothesis. And because of this, we could go to the next step, and the next step again is to do spatial omics in this tissue and try to understand what are the pathways. This is the most important, at least in this particular kind of work. And what we found so far, and these are preliminary studies, we're still analyzing them, but I want to call attention. The first role is amnestic AD, and the second is atypical. So we are seeing much more of these symbols in the amnestic. I mean, we can go in details what they are, but the dysregulation here is much more clear in the amnestic cases than in the atypical case. So again, bringing together this idea that we can use these systems to try to understand heterogeneity in Alzheimer's disease using AD variants as a platform for this, which is my take-home message so far. I'm sure we'll have much more results soon, but I want to call attention just at a recent paper that Melissa Murray led with several people working with post-mortem tissue, how using post-mortem tissue have evolved in the past 10 years, because nowadays we have methods that we can extract omics data and other data from there. So considering this is really, the real model for Alzheimer's disease, and we cannot really model a typical Alzheimer's disease in experimental conditions, I think, embracing this pathomics will be the future for a lot of the things we are trying to do in these diseases. We mentioned the neuromodulatory subcortical PIA. I hope you can join us. We do a lot of things, fun things together with the atypical AD. And of course, thanking everybody who have been generating this data, my partners at UCSF and now at Mayo Clinic. It's really a pleasure to be here. And if you have any questions, this is my email, or I'll be around. Thank you.
The floor is open for questions. And while people are coming to the microphone, I have a question for Leah. It's more of a concept question. From the pathology view, when you mentioned there's more co-pathology in typical Alzheimer's disease and less in atypical Alzheimer's disease, would atypical Alzheimer's disease then more be typical Alzheimer's disease from the pathology?
Yeah, that's a good question. It's a good conceptual question. Actually, it's not that atypical have less pathology. It doesn't have more. Okay. And vascular, at least in our cohort, it's more present in the typical one. So, again, it comes to the question, what is Alzheimer's disease? Is really a disease or a pathogenic confluence that we are seeing different disease conditions with some similarities, a lot of differences? And the next question is, how important are the similarities for us to be able to treat the disease, right? And impede this too. Yeah.
Thank you.
I have a question from Niels Renner.
First of all, thank you for the panel, for all the great talks, and also I think very necessary to have an atypical session every year. Every year they are in my opinion, the best session. First of all, for Deepti, I think we both agree that one of the most important and difficult points to assess atypical AD is at the MCI stage, where it's also very difficult to do these clinical characterizations. And I think work like yours is very important to do this, but in your study, you looked at already clinically defined subgroups. So that tells me then that there is already a clinically variance enough to classify this. So what if we go earlier with your test? Would it be sensitive enough to disentangle these subgroups?
I love this question. Thank you so much, and it's such an important question to ask as we move forward. So I think the big issue here is that by the time people are showing up for clinical presentation, they already have saturated networks. You know, the occipital cortex by, for example, in PCA, is pretty saturated. So they're going to already have secondary domains of impairment by the stage of MCI, certainly at mild dementia. So we do need more sensitive tests in order to detect these, especially at the preclinical stage. I encourage all of you to come to the perspective session later this afternoon, and my colleague, Dr. Rizai, has done some excellent work looking at speech and linguistic patterns just in kind of a couple of minutes of unconstrained and cued speech. Things like that at the preclinical stage, we can really screen out and be able to bring these people to diagnosis earlier.
Thank you.
I know that there's 2 questions from the floor. I'm going to take one question from online, if that's okay, and I'll encourage people who are joining online to continue submitting questions. This is a follow-on to Niels's question for Deep D. Have you compared the utility of your binding task to more standard memory measures?
Yes, thank you. I didn't include that in the talk, but in terms of other validation to memory, other standardized memory tests, is that the question? I think so. Okay, so yes, we actually did find increased sensitivity from this object location binding test. Of course, these different stages of memory are highly correlated with more standard tests like wordless learning tests and even a face memory test. So encoding is correlated with encoding, recognition discriminability similarly correlated with performance on the more traditional tests. But the object location binding is better able to differentiate between the different syndromes compared to the traditional neuropsych test.
I saw a question from Yuta Katsumi.
Great talks. My question is for Rosalina. Super interesting findings, I love it. But I noticed that much of your results are based on ADNI sample exclusively. And I was wondering if you think you would get a different picture once you start to account explicitly for things like early onset and non-amnestic individuals exhibiting non-amnestic syndromes.
Yeah, great question. Thank you so much. Absolutely. I think we need to pool data across cohorts to really get a broader and in-depth picture of what's going on in the broader spectrum. It's true that I showed mostly ADNI data today, and we have also a couple of studies ongoing right now where we are looking at ADNI, JADNI, AdNeuroMed, ABLE, just to get a broader spectrum of all types of AD cases. And there we see also, and there we are trying to build, for example, a longitudinal model of subtyping. So there we see, for instance, there are 2 dominant pathways. One is more of a mediotemporal atrophy pathway and one more of a cortical dominant pathway. And we see, for instance, that some subtypes tend to change allocations over time. So absolutely, more data sets, more diverse populations is absolutely needed, and I think we should move there. But yeah, longitudinal multimodal models are going to be really, really helpful.
Thank you.
A question from Angelina.
Yes, thank you. That was a wonderful session. I feel like I've learned so much actually coming here. My question will be to the speaker from Barcelona. So when you compared these early onset and late onset AD groups, obviously in terms of sleep and neuropsychiatric symptoms, I'm wondering what are the factors that you corrected for, and if there's any confounders that can be explaining some of what we observe?
In regards to neuropsychiatric symptoms, this was a previous study, and at that stage we corrected by SEGs and the prescribed medications specifically, and also the disease stage, so to make sure that if there were MCIs, mild dementia, or moderate dementia, we were taking this into account. And in the preliminary data that I show currently, we didn't correct. It was just a group comparison because we didn't see any differences in the demographics or clinical things that we evaluated, not in the prescribed medications or the clinical phenotypes or the sex difference or others. But the more, now this is a work in progress and we are incorporating more participants. Once we reach the final requirement, we will take this into account in the case that we found any differences, of course.
I'm
Well,
asking
thank you.
because I'm wondering if at baseline some people maybe had poor mental health, so potentially that could be interesting to look at. Thank you.
Thank you so much.
I'm going to ask a follow-on question to Angelina's from online. So first of all, do you look at stratifying for sleep quality or sleep patterns by age? You noted again clearly age effects on some of your regional imaging measures. Do you take a similar approach to look at your sleep measures? And the other question was, what's the effect of sleep apnea on the amount of REM sleep in your studies? What's the chicken, what's the egg?
In terms of sleep apnea, in our sample, we have very little sleep apnea, and most of them happened in late onset Alzheimer's when we compared, not in early onset, but was the early onset AD the ones having less sleep drive. So the problem would be if the sleep apnea was more frequently in the group of early onset Alzheimer's, that it might justify the sleep disruption. But given that, so the conclusion would be even that the sleep apnea is more frequent in late onset Alzheimer's, we're seeing more sleep drive problems in the early onset ones.
Great, I think we have another question from Niels.
Yes, so I have a question for Leah Greenberg. You showed a lot of results, so it was a little bit fast, but you showed some differences in information and metabolic pathways, and then in the end, you showed some differences in dynamic expression, and I don't do any research on this, but it's a very interesting question to me is, what about the ability of these cells in the atypical cases, or perhaps this makes a difference, to cope with the pathology and rewire their metabolic pathways, for instance? I would like to know your opinion on this, basically.
Yeah, I think we have this question also. As we start to incorporate omics in our studies, things become very complicated, right? So we are trying to take an approach of being more hypothesis-driven and to analyze this data, because, again, it's very confusing. So there is a question of compensation. I think it's part of what we are talking about. There is a question is if the selective vulnerable neurons are the same in atypical and atypical. I didn't show this data now, but they seem to be similar. And then, by knowing this, the next step, especially looking at single cell data, is to get these specific vulnerable neurons in these different variants and understand for these particular ones what are the vulnerable pathways or the pathways that are dysregulated. And very important is the glial states surrounding it, which seems to be very different in these two variants. So I think we are still starting to discover. I more brought it to hopefully show some enthusiasm for studying these particular systems and the idea of a typical AD in a way to understand heterogeneity. But maybe something we can do together. I have too much data for just you.
Perhaps it's even not within the cells, but the metabolic environment and availability.
And on that note, we have a related question online. So again, this is regarding Leah making this compelling case for considering differing subcortical vulnerability, even these canonical neocortical characterized presentations. The question is, this change in subcortical activity, would you consider this to be more a hyper or a hypoactivity, which seems to be conferring this more distal neocortical vulnerability?
Yeah, this is a question that's easier to answer in our cohort that I didn't bring here, which is progressive Alzheimer's disease. And again, unfortunately, we don't have early stages of Alzheimer's disease that we know they're atypical. Usually they come to autopsy later on. But we do have for general population studies progressive Alzheimer's disease. And what we see is that for this particular neuromodulatory cortical nuclei, we have an initial lost of some of the subpopulations, and then the remaining populations have hyperactivity. So a good example is the work we are doing over exergic neurons, who apparently is the first population to die in Alzheimer's disease. So what we are seeing is that comparing BRCA0 to BRCA1, there is 40% loss of the number of exergic neurons, but the remaining one, they become very hyperexcited, and this is why we see more exergic in the CSF, despite we have this loss. So we are trying to understand, again, what is compensatory effects and how we study them in statistics, right? This is complicated, and I don't think I have the good answer for this, but the answer we have, yes, there is hyperactivity, at least for a while.
And while I'm not a neuropathologist or physiologist, even the notion of compensatory, it could be maladaptive, for instance. I'll take another, oh, sorry, Neil.
Very short question for Rosalina. You showed very strong results with CAA, but do you have CAA type differences? Can you look at CAA type 1, CAA type 2, or do you not have this information?
No, unfortunately, in ADNI, we don't have that, but that would be great to look at the differences, yeah.
Thank you.
And to ask a provocative question relating to that. So something that jumps out, Rosalie, you did this great job of noticing some of those discrepancies in some of the radiological defined subtypes, whether that's based off modality or at the individual level, right? One discordance I noticed is between some of the data-driven work suggesting the increase or overrepresentation of concurrent alpha-syn in the hippocampal sparing subtypes, and actually what, for example, Leah presented, right? I think that's emerging from other centers like UPenn, like UCL. What do you think is accounting for this discrepancy? Is it like a methods issue, something else?
I don't know. I don't have a good answer to that, but I know that in at least when we have looked at the MRI-based subtypes to FDG-based subtypes comparison, when we look at alpha-synuclein just based in amygdala, for instance, we see more of it in the cortical predominant subtype. It probably also depends on where in the brain you are looking for alpha-synuclein. Then when we look at neocortical alpha-synuclein, we don't necessarily see these associations. So there, I think regional vulnerability to co-pathologies is going to be super important. So yeah, looking at regional versus global, how crude the scales are, it could be a regional issue there.
Leah or Byla, would you like to weigh in?
Well, yesterday I've learned that FDG PET could also reflect astrocytic reactivity. And so I think what you're doing is really great work to compare these modalities, because we assume it's neurodegeneration, but if they show differences, how can that be? So I think, yeah, there's a huge opportunity for these neuroimaging to really look into the pathology. What do they reflect? Is it co-pathology, astrocytic reactivity, which is of course also different with co-pathologies. So I just want to, mention that. Really great work.
And I want to also add to this. I mean, if we have co-pathology, we will have worsening of function in a regional area that you have this co-pathology, right? So this is what you are detecting very well with these different modalities, which is different. I mean, the study that we did, we wanted to know if PCA, are they PCA because they have whatever co-pathology, but they have to meet criteria for PCA. This is not what you're doing, right? You are seeing if it modifies the severity of certain symptoms or regional symptoms. So there's a lot of things to embrace in atypicality in Alzheimer's disease.
May I also ask you, Rosalind? Excellent work, by the way. Have you, within the vascular factor that you're analyzing, have you measured the potential effect of perivascular spaces as a proxy of the lymphatic system and see whether the lymphatic dysfunction might be contributed to heterogeneity?
Yeah, great question. In the study that I showed, we didn't have that measure, but we are working on a study where we are looking at different in vivo markers of cerebrovascular pathology, including enlarged perivascular spaces.
Exciting.
Yeah.
We have a question from the floor, but I'm going to take one from online quickly, if that's okay. So this is from Ryan Townley from University of Kansas. This is more clinically focused. We see this preferential response to acetylcholinesterase inhibitors in young onset, especially dysexecutive cases. Given average delays in diagnosis of perhaps 4.5 years, could perhaps the faster progression in atypical cases be slowed with proper treatment and earlier diagnosis? I presume with access to acetylcholinesterase inhibitors. I think this is probably for Leah and Byla.
Yeah, I think, yeah, Baila, do you want to answer because it's the cholinergic difference that you found in your lab?
Well, I would actually, I have more questions about what do you guys think that explains that difference? Why is the locus coeruleus more vulnerable in these younger ages? What are your thoughts about it?
We know that the locus coeruleus and all the nuclei that are interconnected within the brainstem are by definition vulnerable because during the development they structure differently. They have very tiny nucleus, very long projections with lower myelin, and they also receive very vascular packages. So all the toxins we know that can be more easily accumulated in locus coeruleus. So by definition we know that they are vulnerable, these nuclei, but we don't know why LC is more vulnerable in Alzheimer's disease rather than other nuclei and other pathologies. Because there are other pathologies accumulating tau in the locus coeruleus, but in Alzheimer's disease, the neurons are dying more frequently and rapidly in the early stages. That's something that Leah demonstrated with her work. Maybe
Yeah.
you have some other
I think maladaptation, it's part of it. One very recent finding we have, and it's very intriguing, we separate our results on BRAC stage, right, progressive BRAC stages, and then what happens if you have progressive amyloid stage. So let's say we measure 10 cases that are in BRAC1, and they have different levels of amyloid, right, not in the nucleus, around the brain. So we hypothesize that if they have higher tau phases, it will have less neurons. But actually you're seeing the opposite. So there is a question there, again, it's just an hypothesis right now, that especially in these early stages when tau pathology is starting, because it starts before amyloid, maybe amyloid could be protecting some of these neurons. And this is something we started to study right now, at least at these early stages.
Because the locus coeruleus is also very important in neuroinflammation, and could it also be something glia that is the interconnection between the tau and the amyloid?
It could be, but in this early stage, we are seeing more metabolic dysfunction than inflammatory dysfunction.
Yeah, okay.
Apologies to Rani for joining online for the not straightforward answer, as I understand. But I think we're all behind earlier diagnosis for people with atypical forms. We have a question from Dr. Colin Grote.
Yeah, I'm switching gears. I'm Colin Groot from Amsterdam Alzheimer's Center. My question is for Rosalina, and I was particularly struck by the differences in subgrouping across the different methods that you analyzed. And I was just wondering whether you could speculate on whether that's because the fact that we are subgrouping and we're not looking at it as like in a spectrum level quantitatively, or whether these different people would actually have different, we see different patterns on the scans themselves. So is it a factor of the subgrouping, or is it maybe some other methodological issue?
Yeah, great question. I think we've looked into it, trying to understand what may be the reasons for this, and we think just the nature of the methodologies that identify subgroups are so different. Some of them are just based on visual ratings, for instance. So, and there are others that, for example, only look at medial temporal versus neocortical, certain regions, not the whole brain. Others look at the whole brain, all parcellations. Data-driven methods, for instance, put everything in the model and then let the model decide which regions are important. So I think methodological considerations there really overpower everything else, I would think. So I think that really gives us such different results. But I think if there is a way to maybe benchmark the different subtyping methods, that would be really useful to kind of harmonize how we identify subtypes in the future. Or alternatively, if we move more towards treating heterogeneity as a spectrum, as a continuous process rather than subgrouping, that would also be great, like you suggested.
Thanks, and I agree.
We have another question for Leah. Have you got any data on thalamic activity in typical versus atypical Alzheimer's disease?
No, we wish to go to thalamus in the future, but so far hypothalamus and brainstem.
That's what certainly comes to mind, some of the thalamic nuclei, particularly the lateral nucleus.
Absolutely. You know, we have an R1 who got an excellent score, and I didn't get a notice of award because we have some issues in the US. So, you know, maybe if they release the money, we can look into this.
Well, this relates to maybe my lack of sleep, but something to do with that. And this next question is about deep D. Did you look at the quality of sleep in your patient group in relation to their amnestic profiles?
This is a really good question, and especially the work that we've heard from the rest of the panel. I think this is an important next avenue, is understanding the contributions of not only sleep, but mood. We know that anxiety in particular is highly reported in some of these atypical variants, and indeed across the syndromic spectrum. So, and these neuropsychiatric symptoms have a high correlation with cognitive performance. So we didn't look directly at sleep, but this is really important. Next question.
I also have a question for Deepti. I wondered if you, so you see these differences in the memory across these different atypical subtypes. Do you also see differences in connectivity that explain that for the memory?
This is great. So we did some exploratory analyses. At this point, we don't have a large enough sample size to really answer that question. We did see that the default mode network in particular was strongly associated with storage loss in particular for the objects. And then for location, it was the default mode network atrophy in that network was strongly associated with location encoding and storage. So I think there is something really important about this common network that has this shared vulnerability across the syndromic spectrum. We've done previous work showing a disconnectivity in the default mode network in the atypical spectrum, and we see that it's really the posterior default mode network hubs that disintegrate early with preserved, preserved connectivity to the more frontal hubs of the DMN. So I think we need to study this network more and how it's impacted earlier on, in addition to these initial non-DMN networks.
Yeah. Thanks.
Any other questions from the audience? If not, I'd maybe like to put a provocative question to Leah, who, as I understand it, mentioned at this point, we have limitations in our ability to model atypical forms of Alzheimer's disease under experimental conditions. Have you got any cautious optimism about approaches like induced pluripotent stem cell lines, organoids, or even particular animal models that you think have some opportunities to better get at this selective vulnerability?
Honestly, for this atypical, as we define, these typical forms, I'm not familiar with any successful story of experimental modeling.
Maybe next year.
Maybe.
And on that note, thanks to all the speakers. A round of applause for them, and also for you for your participation.